All right, so glycolysis happens out in the cytoplasm in all eukaryotic cells and also for prokaryotic cells. The rest of the processes in eukaryotes happens inside the mitochondria. Right? For prokaryotes, for bacteria and archaea, this still happens in the cytoplasm. But we're going to focus on the mitochondria. So the mitochondria is a cellular organelle. It's a double membrane structure. Okay, so there's two membranes, an outer membrane and an inner membrane. Each membrane, each of these, is a phospholipid bilayer. Okay, so you have two layers. Phospholipids is just like the outer cell membrane. There are proteins embedded in it as well. So, for example, the pyruvate from the cytoplasm at the end of glycolysis has to get into the mitochondria, and that means it has to cross over two membranes to get through there. So we have to transport that in there with membrane proteins that will move the pyruvate into what's called the matrix, which is the space inside the mitochondria. And that's where the next two processes happen, pyruvate oxidation and the citric acid cycle. So we're just going to go through these as a general outline and an overview of what happens in these two processes. So the pyruvate, if you remember, ends up with three carbons. Again, there's oxygens and hydrogens here. I'm just kind of giving you the basic skeleton of it. The pyruvate gets oxidized, which means that it loses electrons. Those electrons go to NAD plus, making NADH. What also happens during this process is one of the carbons is pulled off into carbon dioxide. And so we end up with just two of these carbons remaining. And those two carbons are then attached to a helper molecule called coenzyme A. So this is then referred to as acetyl, because that's, if we were to draw out the structure with the three hydrogens here and the oxygen and hydrogen there, it's called an acetyl group. And so it's acetyl, and so I'll give you that actual detail I'll add in there. So this actually has hydrogens here and a double bond to an oxygen there. So that's an acetyl group, and it's attached to a coenzyme A, so it's called acetyl, orange here, acetyl-CoA. That's the name of this molecule. So there are three products of the process of pyruvate oxidation. One is NADH, two is carbon dioxide, and three is acetyl-CoA. This process is often overlooked. People often will jump from glycolysis to the citric acid cycle. And one of the problems with that is that glycolysis ends with pyruvate in the cytoplasm, and the citric acid cycle is going to begin with acetyl-CoA inside the mitochondria. So there's kind of a disconnect, right, if you don't cover this process, because we have questions. How does the pyruvate get from the cytoplasm into the mitochondria? When did that happen? So there's a process that has to transport it in there. Where does this acetyl-CoA come from? Well, it actually comes from the pyruvate. So this was pyruvate. And the pyruvate is oxidized and then becomes acetyl-CoA. So that's called pyruvate oxidation. This will happen twice for every glucose. So every glucose molecule that we started with had 6 carbons. It broke down into 2 pyruvates. So this happens twice for every glucose. That means we get 2 of these for every glucose, 2 of these, 2 of these, and so forth. So that's it. That's pyruvate oxidation. You end up with this acetyl-CoA molecule. Again, we're not going to go into all the details of the chemistry, but what we're going to enter now is in what's called the citric acid cycle. Citric acid cycle is also called the Krebs cycle because of this molecule. K-E-T-A-T. Sorry, speaking. Okay. So we're going to start now the citric acid cycle, also called, I'm not sure if I can figure out a way to not get that to squeak. Okay, Krebs cycle. So it's called the Krebs cycle because of a person named Hans Krebs who figured that this process was actually a cyclical process as opposed to a linear pathway, like glycolysis. Many biochemical pathways start with one molecule and they end with another. And then there's intermediate molecules in between. And what you end with is something very different than what you started with. what people found when they were studying this next process was similar to everything else. You start off with some molecules, they change, they're transformed into new molecules, different molecules. But at the end of the process, they were finding concentrations of this chemical, this thing called oxaloacetate, to be unchanged. They're the same as they were in the beginning. So they would add oxaloacetate to acetyl-CoA and then get, with the two of them combined, a 6-carbon molecule called citric acid. Again, this is just a carbon skeleton. It's not representing all the oxygens and hydrogens. You don't have to know that right in this class. So eventually what's going to occur is a whole series of reactions. And I'm not going to go through all the reactions. That's not really something that we need to know right in this particular class. But we're going to go through some of the events that happen during this process. So some of the events that happen during the process are that this molecule here, the citric acid, is going to become oxidized a couple more times. So again, this whole process is all about oxidation, 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 which is all about removing electrons, right? So electrons going from one molecule to another molecule who's the electron carrier. That's really the whole point of all of this. Glycolysis, pyruvate oxidation, citric acid cycle. It's to make these. That's really the main thing. So that's going to happen here. And what's going to occur as those events happen, as the oxidation event occurs, I'll try to keep it consistent with color here. So 1, 2, 3, 4, 5, and then a carbon dioxide. So one of the carbons gets cut off, essentially. An oxidation occurs, and you'll make NADH. The same thing will happen again. Another carbon dioxide will get cut off, another oxidation event. Right, that'll give off another carbon dioxide. And when that happens, you get another NADH. Now here, if we're not looking at details, we're just looking at super generic structures. 4 carbons there, 4 carbons here, kind of the 2 carbons of the acetyl group gone as carbon dioxide. So that would seem like the end of it. We have completely oxidized the original 6-carbon glucose. And all of its individual carbons of the glucose have now been lost as carbon dioxide. That was part of that original formula, that the 6-carbon glucose goes into the 6 carbon dioxide molecules. And that would have happened now if this process occurred twice. This happens 2 times, once for each pyruvate. One pyruvate, once for each acetyl-CoA. So one acetyl-CoA, then a second acetyl-CoA. But that's not the end of it. Okay. What happens is that this molecule, that's what makes this unique, and this is what Hans Krebs figured out, that this molecule here gets rearranged back into oxaloacetate. And instead of that costing energy, which you would think, like, so it costs energy to make these changes, it actually allows additional oxidation events and more energy to be pulled out of the process. So when this 4-carbon molecule is rearranged into another 4-carbon molecule, and then into another 4-carbon molecule, and then back into oxaloacetate, we can actually have an extra oxidation to make one more NADH. We could get a different kind of oxidation to call something called FADH2. Don't need to know the details of that. It's another electron carrier. It's something just like NADH. It takes away 2 electrons from the process, stores energy. It's going to be able to be used to then make ATP later. So it's important. And in addition to all that, you get a phosphorylation. So the production of most books in the simple, like the non-majors textbooks, are going to say ATP. It's really GTP, but we often will refer to it as an ATP molecule. Or essentially, a phosphorylation takes place, and we make one of these molecules by taking, just like in glycolysis, where a phosphate from the environment comes in, is attached and removed again, and then it can be added to this molecule. All that stuff happens as it goes back to oxaloacetate. And Hans Krebs figured that out, that it was a cyclical process to regenerating that molecule as it does a whole bunch of extra things. So this is a much more complicated process, has many, many more steps into it. But what you really need to know, so what do you need to know about the citric acid cycle? So the citric acid cycle produces from the end of the so what comes into the cycle, I guess we should say, is acetyl-CoA, and we need this oxaloacetate. All right, what comes out of the process? So what are the products? And these are all produced inside the mitochondrial matrix. So this is all happening inside the mitochondria in this large space, or for the mitochondria, it's large. So this is the space inside to the interior of the mitochondria. What we're making from the process, so the products are 2 carbon dioxide molecules, 1, 2. We're going to get 3 NADH molecules, 1, 2, 3. That's per turn, of the cycle. So we'll come back to this in a second. You're going to get 1 of these things called FADH2, which is pretty much the same. So you can kind of lump these together for now. And we're going to make 1 GTP, so which is just, again, the same as ATP. And it regenerates oxaloacetate. All these products would be times 2, right? Because for every 1 acetyl-CoA, this is what would happen. But there are going to be 2 acetyl-CoAs because there were 2 pyruvates from each glucose. So for every 1 glucose, this will be adding up. So essentially, for every glucose, there'll be 4 of these because you would have had 1, 2 from 1 acetyl-CoA, 3, 4 from a second acetyl-CoA. All these things happen twice, so that's actually 6 NADH and 2 of the FADH2s. That's 2 of these GTPs, and then obviously 1 oxaloacetate per cycle. So again, 2 total are being made here. So that's just a little summary of pyruvate oxidation and the citric acid cycle and the events that happen there, which again are largely oxidation events. What we're seeing, though, is we haven't still made a lot of ATP. So every glucose molecule, so we start with glucose. Each glucose is responsible for generating between 36 to 38 ATP, and I'll talk about why there's a little bit of a difference in the next step. How many have we actually made up to this point when we look at glycolysis, pyruvate oxidation, and the citric acid cycle all combined? So we combine all 3 of these processes, and what have we made? Remember, you gained 2 from glycolysis. There are none in pyruvate oxidation, and you get 1 per turn of the citric acid cycle. That would be 2 there. That's just a total of 4. So it means at the end of all these, we've made 4 of these molecules, of the ATPs. We still have 34 more to go to show how they're made. Right? But we're done with this. So where's the energy? It's all locked up in these molecules as the electrons. So that's our next step now, and our final step of this is to look at what's called the electron transport chain and how this is then converted from electrons into concentration gradients, and then how that's used to make ATP. And this, what the importance of this, is that it combines multiple concepts that are part of this course. It's going to combine membrane structure, membrane transport, diffusion, and enzyme activity, and the energy being transferred, from a reaction through use of an enzyme. Okay, so it's going to combine all these things into the process that we're about to go into next.